thank you for joining us. Yes, now I would like to give time to our principal, Professor S.T. Genesius, for the inaugural speech.
Good afternoon. I'm not a chemist to speak much about asymmetric synthesis. Even though I studied chemistry in the school level, when I turned to college, I moved from science to arts. So I'm not very familiar with the asymmetric synthesis and how it is helpful in chemistry, and modern day chemistry is completely depending upon this kind of synthesis. I know that, but I'm not very familiar to talk concretely on this issue. But however, what I would say as a inaugural speech is that these talks will enlighten students in a better way to know the subjects. So that is my wish, and that is how I look at it. Every guest lecture enlightens the students in a particular topic and gives a new view of reality, what they learn in the classroom, with connecting it with real life situation. So that is more important. Otherwise, the learning becomes meaningless sometimes, because bookish knowledge, it remains book, and they do not know why they are studying these things. Now, these kind of lectures will give them some insight, so that what they learn in the classroom manipulated. This is how day-to-day chemicals are being prepared, or it is used in different fields in different way. So this kind of application-oriented knowledge is very much needed, and such a knowledge they can pick only from these kind of lectures. Otherwise, classroom lectures will remain classroom lectures, because they are focused on syllabus and the main topics. So I always encourage the departments to come forward to organize these kind of lectures, so that the students will get a different view of reality than what they learn in the syllabus. So in that way, I appreciate the chemistry department for taking initiative on this one and inviting the research scholar to give a orientation to our students, give a lecture on this synthesis. That is modern day. It is completely, chemistry is completely depending upon that, I know for certain. So I welcome her once again, and also I congratulate the chemistry department, Ajam and Rahul sir, for taking pains for organizing this kind of program for the students. And also I request the students to make use of this opportunity to learn, to ask some questions, so that you know why you are learning a particular subject in a paper, and how it can be applied in real life situation, and how you can become a chemist or a scientist in the future, so that you can also involve in this kind of new inventions and different combination of chemicals, so that it will be useful for human beings in different ways. So with this, let me wind up my talk and give the students more time. Thank you very much.
Thank you very much, sir. Thank you. Okay, without further ado, we will just go on to, I'll give the time to our resource person now. Please take your time.
Thank you very much, Ms. Ajam, for kind words of introduction. And I would also like to thank the principal and the Department of Chemistry for inviting me, and also the organizing team for giving me this privilege. So let me share my screen first. Okay, so today I will discuss about my research topic, that is on asymmetric synthesis. But before that, we will just look into some basics of this stereochemistry, which we need to know in order to understand what is asymmetric synthesis. So see, as we are not going to be able to discuss all the cover all the topics, I will just give you a peep into this important subject that we have before us today. So these are just some basic terms to remember. So I will start with the isomers. So these isomers are ions or molecules with identical formula, but different structural or arrangement of atom in space. So we have two major classes of this isomer. The first one is our constitutional or structural isomer, where this type of isomers will differ in the way the atoms are connected. That is, this type of isomers will have a different structure, and they have different IUPAC names with the same and different functional group and different physical and chemical properties. These IUPAC names, these are the names of organic compound as recommended by the International Union of Pure and Applied Chemistry. And the second class is our stereoisomer, where this type of isomers, it will differ only in the way the atoms are oriented in space. That is, it will have different configuration or this 3D arrangement, and this atom will have identical IUPAC names with different prefix, and they have the same functional group. These prefixes are just a group of letters that are added before the beginning of a word. So this is an example for this constitutional isomer and stereoisomer. So see, in our constitutional isomer, we have two molecules with same molecular formula. But the first molecule, see the left-hand side molecule, we have our methyl substituents, which is connected to carbon number 2. And the right-hand side molecule, we have our methyl substituent at carbon number 3. So although they have the same molecular formula, these two compounds have different structure. Therefore, they are called as a constitutional isomer, and this will have different IUPAC name as 2-methylpentane and 3-methylpentane. And the other, this is the example of stereoisomer, where we can clearly see that we have two molecules with different configuration. See, the left-hand side compound, we have both the methyl substituents which are above the plane, and the other one have one methyl substituent above the plane and one below the plane, although they have the same molecular formula. So this type of compound, they will have the same IUPAC name as 1,2-dimethylcyclopentane, but they will have different prefix as cis and trans. So this is the flowchart through which we can figure out what type of isomers our compound possess. So first, you have to check whether your compound has same molecular formula or not. If yes, it is an isomer. And under this isomer, again, we have two classes, constitution isomer and the stereoisomer. So if our compounds have the same connectivity or same structure, they will come under this stereoisomer. And if not, they will come under this constitution isomer. So under this stereoisomer, see, we have again two different classes. First is the configurational, and the second one is conformational isomer. So if our compounds can be converted by rotation about this carbon-carbon single bond, then this will come under conformational isomer. And if not, they will come under this configurational isomer. Under configurational isomer, again, we have two classes, geometrical isomer and the optical isomer. So this geometrical isomer, these are based on their 2D structure, and this optical isomer, it is based on their 3D structure of the compound. And under this optical isomer, again, we have diastereomers and so these two we will discuss in more details in the upcoming slides. So see, under constitutional isomers, these are the isomer with same molecular formula but different structure. So we have 4 different classes under this isomer. This first one is the chain isomer. So this type of isomers will have different connectivity or different number of carbon along the carbon chain. So see, I have given here 3 molecules. So see, in the first molecule, we have 5 carbon in the carbon chain. In the second one, we have only 4 carbon in the carbon chain. And the third one, we have only 3 carbon in the carbon chain. So this type of compounds are called as chain isomers. And the second class, we have the positional isomer, where this type of isomers will have the same functional group and the same molecular formula, but this functional group will be located at different positions along the carbon chain. So see here in example, we have 3 compounds with same molecular formula, but see the alcohol functional group is located at carbon number 3 in this first compound. And in the second compound, we have this alcohol functional group at carbon number 2. And the third compound, we have alcohol functional group at the carbon number 1. So this type of compound are called as the positional isomers. The third one is our functional group isomer, where this type of isomers will also have the same molecular formula, but they will have different functional group. So let us see the example here. See, I have 2 compounds here with same molecular formula, C2H6O. But see, the compound A have alcohol functional group and the compound B have ether functional group. So this type of compound will come under this functional group isomer. And the last class, that is the metamers. This type of isomerism, it is due to this unequal distribution of carbon atom on either side of the functional group or the heteroatom. So see here in this example, I have two compounds with carbonyl functional group. So in first compound, let us see, we have only one carbon atom on the left-hand side and three carbon atom on the right-hand side. But in the second compound, we have two carbon each on both sides. So this type of compounds are called as metamers. Now coming to geometrical isomer, so we know that a double bond is made up of a pi bond and a sigma bond. So this pi bond in the double bond, this will not allow the rotation of the molecule such that all these group attached to this alkene, they will lie on the plane. So let us see this example here, butane, where see all these group attached to this alkene will lie on the plane. And if the group attached to this alkene, if they lie on the same side, see in this case, we have both the substituents position on the same side. So this molecule will be called as cis molecule, and the other molecule where the substituents are on the opposite side, it is called a trans molecule. And similar example here I have given where our more complex molecule having higher priority group, that means atom with higher atomic number. So see in this example, between the hydrogen and chlorine, the chlorine have higher atomic number, so it will be given the higher priority. And in case of this, hydrogen and bromine have the higher atomic number, so it will be given the higher priority. So since both the higher priority group are on the both side, this will call as cis molecule. And in this case, we have this chlorine and bromine on opposite side, so this molecule will be called as trans molecule. So these isomers are also called as cis and trans isomer. And we also observe that these cis isomers are usually more unstable than the trans molecule. This is because see, in the cis molecule, we have both the bulky substituents on the same side. So there will be steric repulsion between these two groups. So it will be unstable comparing with the trans molecule where no such repulsion exists. And similarly, see the compounds with bonds in ring. If our substituents are on the same side like this, it is called a cis molecule, but if the substituents are on the opposite side, it will call as trans molecule. And take a note here that if one of the two carbon atom of a double bond has two identical substituents, like this compound here, 1,1-dichloroethane, where this carbon have the two identical substituents, so this type of compounds will not have cis and trans isomer. Now coming to chirality, so these chiral objects are those with left-handed and right-handed form. So I have given here example, our hands and ears. So see, we can say that our right-hand side is the mirror image of our left-hand side. And when you place your right-hand side above your left-hand side, you can clearly distinguish between these two hands. This means that the mirror image is not same with the image itself. So this type of objects are called as non-superimposable mirror images. And achiral objects are those whose mirror image is same with the image itself, so they are superimposable mirror images. Example of achiral object here I have given chair and spoons, where we can clearly where the mirror image and the image is indistinguishable from one another. So these type of objects are called as chiral objects. So let us find out about the molecules also. So see, these molecules, like they are also like these chiral objects that have left-handed and right-handed form. So these handed substances are said to be chiral, and these molecules that are chirals are now superimposable to the mirror images like the chiral objects. Now coming to chiral center. So see, chiral center, it is a point in a molecule where 4 different groups are attached to a carbon. So see, in this case, in this molecule, this is the chiral center where 4 different groups are attached to it. So in modern terms, this chiral center is also called as stereogenic center or the asymmetric carbon. So if a molecule have only one stereogenic center, they will always be chiral. But if a molecule have no chiral center, then it will not be. It will always be achiral. And in case when our molecule have two or more chiral center, then they may or may not be chiral. So let us see the example here. I have given cis-1,2-dichlorocyclopentane. So see, in this molecule we have two stereogenic center in this molecule, these two carbon. But we also observe that there is an internal plane of symmetry in this molecule. That is, see, the molecule is a mirror image of itself. So this type of molecules are said to be achiral and are called as meso compounds. Now coming to enantiomers, so these enantiomers are those which are non-superimposable mirror images, and they will be different molecule. And the cis isomers will always be achiral, and the trans isomer will always be chiral. So let us see the example here. I have given the cis molecule of 1,2-dichlorocyclopentane, and this is the mirror where we see that the mirror image is same with the image itself. So they are superimposable mirror images. Therefore, they are same compound and they are not in. But in case of this trans molecule of 1,2-dichlorocyclopentane, see, the mirror image is not superimposable on the image. So these two are different compounds, and this type of compounds are called as enantiomers. And I have another example here, an open chain compound where our mirror image is where our mirror image and the image are not similar. So these two compounds are also called as enantiomers. And see, since our enantiomers are different compounds, they need to be distinguished by name. So these are done by adding the prefix R and S before their IUPAC name. And the naming of this enantiomer is called as Kahn-Ingold-Willoch system or the CIP system. So see, according to this rule, first we have to rank all the atoms attached to this chiral center according to their atomic number. So see example in this molecule, and note that the highest priority will be given to the atom with highest atomic number. So see, in this molecule we have bromine as the highest priority group, followed by chlorine, carbon, and then hydrogen. After ranking this atom, you make sure that your lowest priority group, that is the hydrogen here, this is placed at the back of the molecule. After that, you trace the path of the first three ranked substituent. So in this example, see here in compound A, when you trace the path of the first three ranked substituents, it follows a clockwise direction. So this chiral center will be assigned as R. And in the example, see here in B, when you trace the path of the first three ranked substituent, it follows an anti-clockwise direction. Therefore, this chiral center will be assigned as S. Now coming to diastereomers, so these diastereomers are stereoisomers which are not mirror image of one another. So these are the stereoisomers which are not enantiomers, and these diastereomers are molecules with two or more chiral carbon. So let us see the example of diastereomer. So here we have two compounds with similar structure and same molecular formula. But see, this right-hand side compound, this is not a mirror image of this left-hand side compound. So these two compounds are known as diastereomers. And also note that all the configuration of the chiral center in diastereomers are not inverted. So see, in this case, the configuration of this chiral center of this diastereomers is S and S, but the other one have S and R. The first one remains as same, but the second one is inverted. But in case of enantiomers, this configuration of this chiral center, it will always be inverted. That is, if one of your enantiomer have SS configuration, then the other one will have RR configuration. And naming of these diastereomers are also done using this Kahn-Ingold-Willoch system. So now let us understand how this chiral molecule react differently with this biological molecule. So see, as we know, most of the building block in our living system, they exist as enantiomer. That is, this protein, DNA, and this amino acid, all these exist as a single enantiomer. So when this biologically active compound, that is our chiral when it will react with this biological molecule in a chiral manner. So see, when our chiral molecule, when it does not fit to this binding site of the receptor of this biological molecule, this will not give any response. But if it fits to this binding site of the receptor, then it will give a response. So this 3D arrangement or this configuration, this is very important, especially in the field of pharmaceutical, as the slight difference in their 3D spatial arrangement can make a difference between the targeted treatment and the undesired side effect. So many of the transformation that you encounter, it will have the potential to produce multiple type of products. So depending on this product form, let us see what are these stereoselective and stereospecific reactions. So see, I have given a general example here of stereoselective reaction. The stereoselective reactions are those reactions which produce one stereoisomer of the product preferentially over the other. So see, in this case, we have compound A and B which will react to give product C and D. So if these two products are formed in unequal amount, it is called a stereoselective reaction. And if the two products are enantiomer, it is called an enantioselective reaction. Or if they are, if these two products are diastereomers, it is called a diastereoselective reaction. So see, here I have given an example of enantioselective reaction where an aldehyde will react with dialkylzine in the presence of chiral ligand to give two enantiomer in unequal amount. So let us assume that this is the R enantiomer and this is the S enantiomer. So here we have 70% yield of the R enantiomer and 30% yield of this S enantiomer. So this enantioselective reaction, they are expressed in terms of enantiomeric excess, which is given as EE. So this enantiomeric excess, it is the excess of one enantiomer over the other, which will be expressed in percentage as a whole. So see, in this case, the enantiomeric excess for this reaction, it is calculated using this formula where our major amount minus the minor amount divided by the whole amount into 100. So in this case, we got 40% of enantiomer excess, which means that we have this R enantiomer 40% more than the S enantiomer. And these stereospecific reactions are those which will give only one specific product. So now coming to this asymmetric synthesis, so see, it is clear from the word itself that we are going to generate a molecule which is not symmetric in nature. That means we are going to generate a chiral molecule, a molecule which will be chiral in nature. So it is a chemical reaction in which one or more new elements of chirality are formed in a substrate molecule which produces the stereoisomeric product, that is our enantiomers and the diastereomers in unequal amount. So simply, we can say that this asymmetric synthesis is a type of reaction which produces the chiral molecule in unequal amounts. So these asymmetric synthesis are often known as enantioselective reaction. So you must be wondering why these asymmetric synthesis are important. So let us find out about it. So see, as I've already explained to you before, how this chiral molecule react with this biological molecule. So see, different enantiomers and diastereomers of a molecule have different biological properties. So we find that this chiral molecule, when they have different biological properties when it comes to reaction with the body. So see, as the living system are themselves chiral, so each of the enantiomer of this chiral graph can behave very differently in vivo. In another words, the R enantiomer of a graph will not necessarily behave the same way as the S enantiomer of the same graph when taken in by the. So see, let us see the example. So this example, see this graph. This is a main graph which is used as a racemate, which is given as a racemate mixture in earlier days. So racemate mixture means 50% of this R enantiomer and 50% of this S enantiomer. So this drug was given to pregnant women as a sedative for morning sickness, but this thalidomide causes a severe side effect, that is, thousands of babies were born with missing arm, leg, feet. So in many countries, it was banned by 1961, and later the scientists discovered that it was this thalidomide which causes the severe side effect. And another example, R-naproxen, where the S-naproxen is used as a drug for arthritis, and the R-neproxene causes liver poisoning. So see, if you give a racemate mixture of these two chiral molecules to a patient, the patient may get relief from arthritis, but they may die of liver poisoning. So it is very important that a single enantiomer is made in order to avoid such side effect. So see, before this thalidomide rigidity, almost all the commercial drugs were sold as a racemate mixture that is 50% of R and 50% of the S enantiomer drug. But after this thalidomide rigidity, now scientists started developing these drugs as a single enantiomer, and now many of the commercial drugs are sold as single enantiomer drugs because often one enantiomer exhibits the desired pharmaceutical activity while the other enantiomer is inactive or in many cases even harmful. So now more than 40% of the commercial drugs that we have, it is enantiomer, they are sold as single enantiomer, and it is predicted that 75% of this drug will be sold as a single enantiomer in the coming years. So this laboratory synthesis of this chiral compound from achiral or the racemate starting material, it will always give a racemate mixture of enantiomer. So if you want to make just one enantiomer, you have to use a starting material or reagent, which are also just one enantiomer. So let us understand why we need a chiral component to achieve this asymmetric synthesis. So I will explain this using this carbonyl compound as a reference. So see, in this case, when both our nucleophile and the carbonyl compound, if both our reactant and our reagent, if they are achiral, then there is equal possibility for this reagent to attack from both the sides. So this will lead to a transition state with equal energy, and hence the product will form in equal amount as a racemate mixture. So see in this figure here, when we use achiral carbonyl compound and achiral nucleophile, so the nucleophile can attack from the front side or from the back side. So this will result in equal energy in the transition state, hence the product will be formed as a racemate mixture. But when either one of your starting material or the reagent are achiral, then you will get a product in unequal amount. So in this case, we have a chiral carbonyl compound with a chiral nucleophile. So see, in this case, the chiral center in this carbonyl compound, they will control the energy of the incoming group, such that this nucleophile will prefer to attack from the side where there is less steric repulsion. So this will result in a transition state with lower energy, and hence the product will be formed more easily and in more amount. And similarly, when you use a chiral carbonyl compound with chiral nucleophile, this will also result in a product. This will also result in asymmetric synthesis, where one product will be formed in more amount. So how do we chemists control this chirality? So this is done by using a chiral component as a starting material. So let us see some methods for this asymmetric synthesis. So first one is our chiral pool synthesis, where these are simplest and oldest approach for initial selective synthesis, and it uses an enantiomerically pure natural product as starting material. So see, in this case, we have aspartame. It is a nutritive sweetener, which is made from a starting material S-phenylalanine and S-aspartic acid, which are natural products. And the second method is the use of chiral auxiliary, where this chiral auxiliary are enantiomerically pure compound, and it is attached to the starting material to give one enantiomer of the product. So see, in this case, we first add the chiral auxiliary to the substrate. After that, it will undergo a desirable reaction to form the product. After forming the product, you can remove this chiral auxiliary using a suitable method to get the product as a single enantiomer. So let us see the example for this use of chiral auxiliary. So the example of dialdase reaction between cyclopentadiene and benzyl acrylate. So see, in this example, we have both our starting material, which are achiral, and this group that is attached to the alkene, this will lie on the plane. So there is equal possibility for this incoming group to attack from the back side or from the front side. So it will result in two enantiomer as 50-50 mixture. But see, in the second example, where we introduce the chiral auxiliary to one of the starting material. So see, in this case, in the chiral auxiliary, we have a group that lies above the plane. so this incoming group will not prefer to attack from the front side because there will be steric repulsion from this group. So it will only favor to attack from the back side to get the product as a single enantiomer. So after the formation of this product, you can remove this chiral auxiliary using a suitable method to get the product as a single enantiomer. And our third method, that is the use of chiral reagent. This chiral reagent, they give only a single product. This example of this chiral reagent is binyl H, where our binyl binyl will give the S enantiomer and R binyl will give R enantiomer. But see, although these three methods it give a good enantiomer with excess, it is not a favorable method anymore. This is because see, there are lots of disadvantage. In these three methods here, the chiral pool synthesis, that is the first method, this number of possible reaction the molecule can undergo is restricted, and also lengthy synthetic route may be required, so it is not favorable. And the second method, that is the use of chiral auxiliary. These are not atom economy reaction, as there is an extra step for this addition and then removal of this chiral auxiliary. So it is a time-consuming reaction. And the use of chiral reagent, see, although this method also give a good enantiomer with excess, only a few reagents are successful and it can be used only for a few reactions. So it is not favorable in comparison to the newly developed methods. So now we have the last method, that is the use of chiral catalyst. So see, this use of this asymmetric chiral catalyst, this provide the best method for atom economy. The extra step for the addition and then removal of this chiral auxiliary is removed, and then the synergistic function of this active site of the chiral, this will make the substrate more reactive in the transition state. Hence, the reaction can be complete in shorter period of time. And we have two types of this chiral catalyst. First one is organometallic base, and the second one is pure organocatalyst. So let us see this advantage of this organometallic base compound. So see, the metal can act as Lewis acid and base, hence it can activate the starting material, which will lead to the completion of the reaction in a shorter period of time, and basic condition is avoided. So see, traditionally some of the reactions are carried out in the presence of basic catalysts like the metal alkoxide and hydroxide. So see, this basic catalyst, it gives a good result, but then this will give byproduct due to competing side reaction. And the organometallic catalysts, this work firmly under neutral condition, but there are some limitations to this metal-based catalyst. That is, most of the metals are sensitive to air and moisture, so it requires the exclusion of air and moisture, which is very difficult to carry out. And some of the metal catalysts are poisonous, and some of them are expensive. So these are the real challenges in the preparation of the pharmaceutical compounds. So now we have the most favorable method, that is the use of organocatalyst as a chiral catalyst. So see, these organocatalysts are small organic molecule having the catalytic activity. So the use of this small organic molecule as a chiral catalyst, it have recently been recognized as a valuable alternative or addition to the existing well-established, often metal-based methodologies, driven both by the scientific interest which accompanies the emerging field, and also the recognition of the huge potential of this new era. So the benefits of this organocatalyst are: it is a small, purely organic molecule, it is stable in air and moisture, easy to handle, and can mimic the role of metal as Lewis acid and base. So see, most of the currently used efficient catalysts, they have more than one active site. Almost all the reported catalysts now we have, they act as a bifunctional catalyst. So they can activate both the they usually have this Bronsted acid and Lewis base, so they can activate both the donor and the acceptor. So it not only accelerate the reaction, but they also increase their selectivity due to these highly organized transition states. And the technical application for these organocatalysts are accessibility to both the enantiomers with comparable price, low molecular weight, easy separation from the product, and some catalysts can be recovered after workup without racemization. So see, today, although most of the asymmetric-based catalysts, no, it continued to rely on this organometallic-based compound, but this picture is changing slowly, and this organic catalyst is developing into an important segment in organic chemistry because of their operational simplicity, ready availability, and as an inexpensive catalyst. So currently, even in our lab, we are trying to develop some organocatalysts which can be used for these broad substrates and for reaction, since most of the reported catalysts are highly substrate specific. So this development of new organocatalysts for enhanced reactivity and catalytic efficacy is always important. So say if you are interested to do research work, you can always opt for this field, where you can build this, where you can build the building blocks of these cosmetics, pharmaceuticals, and then agricultural compound. So that's all, and thank you very much, everyone. And here are my lab mates and my supervisor, Professor Ganeshan Base. I thank all of them for their continued support also, and thank you very much, everyone, once again.
Thank you so much, Ms. Mezhübeinuo, for that very insightful presentation. This is really something new for me as well, and it is really interesting, especially because I've been teaching my students also about chirality, and we have done this even in, I think, in the first semester as well. We're studying stereochemistry and all that. So I'm hoping some of my students can ask some questions. You can enter your questions to the chat box, or you can just talk to the resource person herself. In the meantime, I also want to ask you a question like, what is the biggest challenge that you face practically, like when you're doing asymmetric synthesis of any compound?
For me, the biggest challenge will be separation of this compound. It's very difficult because like both these enantiomers, they have almost same this RF. Okay, so it's very difficult to separate this compound. So it takes like 4 or 5 hours even to separate even 10 mg amount like that. So that is the biggest challenge for me.
Okay. Well, how do you separate? How do you distinguish the RF? What exactly? Like what methods do you use?
We run this TLC plate from where we can find out the RF of these chemical compounds. And after that, you can do column chromatography for the separation of the compound, or even there are other methods like HPLC, high performance liquid chromatography, or if in industries there are methods like simulated moving back chromatography, which is recently developed for this industry. So those methods are like it can separate this two enantiomer in a good amount.
Okay. Okay, thank you. As you can hear, class, students especially, like this is the study that she is doing is completely multidisciplinary when it comes to chemistry. So it is something you can also look forward to, like if you ever want to do research or do a special paper in the future. So if any student has any question, please, you can go ahead. Chat box is open.
Yes, if you have any doubt, please feel free to ask. Even me, I'm still at the initial stage of my research, but see the little knowledge that I have, I want to share with all of you. And later on, also, see if you in future also, if you want to know about my work progress or about this research field, then I can give my email ID to the organizing team, so you can take from them, and you can feel free to contact me any.
Okay, I suppose there are no questions. Are there any? Is there any student, especially chemistry honors, if you want to ask any question? Because Ms. has covered a really good portion of organic chemistry, organic stereochemistry to be specific. Okay, I suppose there are no questions. Okay, so like Miss has said, you can like even contact her, and you can ask her directly, or you can contact her through me as well. So okay, I would like to give the time now to Sir Rahul for the vote of thanks. He is our HOD from Chemistry Department.
Yeah, then Srishti is having a question actually. Hello?
Okay, sir. Okay.
Srishti is having a question, like she's asking a question.
Okay, sir.
Can more development take place in the field of biochemistry on the basis of this concept? Yeah, we'll be very grateful if Ms. Mezhübeinuo explained that part.
Mezhü, can you hear us?
Can you please repeat the question, please? It's not very clear. So
Actually, one of our students asked a question regarding more development can take place in the field of biochemistry on the basis of this concept. Can you
yes,
hear
sir.
me?
Yes, sir.
The question is in the chat
See, in the
box.
in
Please check that. Yeah, if you can.
yeah, question in the chat box. Yes, please check.
Can more development take place in the field of biochemistry based on this concept?
Yes, very much, because see, the drug designs, they attempt to use the structure of the protein as a basis for designing new ligand by applying the principle of this molecular recognition and all. So this drug design molecule involve the design of molecules that are complementary in shape and charge of the biomolecular target with which they will interact, and therefore they will bind to it. So so you got you got me.
Srishti, you can talk directly to ma'am, to Miss. Okay.
See, these drugs are designed, no, looking at the principle of the molecular recognition. So it is very important that we know this binding site of the molecular biomolecules also, right? That's why this method, I think it's very important, since this drug design will based on applying this principle of molecular recognition. So I think it's very important that we know the shape of this receptor site of this biological molecule first.
Okay. Okay. Thank you. Okay. Anyone else has any question?
I think Ms. Mezhübeinuo will be very happy to take 1-2 questions.
Yes, sir.
Okay. You have given such a nice explanation that I think it's clear for everyone. But thank you so much, Ms. Mezhübeinuo, for your quality time, and you explained such a wonderful, such a typical concept of organic chemistry, where every time students needs repetition, I mean repeated explanations, and you covered almost each and every topic under this, each and every concept under this topic, like type of isomerism, geometrical isomerism, stereoisomerism, diastereomers, enantiomerism. More importantly, the concept of superimposability. It's so nice presentation. Thank you so much for giving you quality time. I mean, when the situations become normal, I mean, after this pandemic COVID-19 situation, surely we'd like to invite you to our college for giving a offline presentation. We'll be very happy and we'll be very grateful if you accept our invitation in future. And apart from that, I'd like to thank Sir Principal Sir, for a nice inaugural speech. I would like to thank our administrator, Sir Peter, for giving the quality time and going to our webinar. And I would like to thank all our students for participating and going to the webinar. And this concept, I mean, whatever our guest explained, that is a part of our organic chemistry syllabus, particularly in the second semester as well as fifth semester. So in the second semester, especially for first semester students, the next year it will be each and every topic, each and every concept will be very, very helpful for them. And I can find some research scholars, especially Mr. Wasang Lang from NEHU, is a research scholar from NEHU. Thank you so much for giving you quality time. I can find some our research scholar from this NEHU, Nagaland University as well. So thank you so much for everyone for giving your quality time. And thank you, Ajahn, for arranging. Finally, thank you, Ajahn, for arranging this kind of webinar in such a situation when we are struggling with a very shortage of faculty members. We are only 2. We are really struggling with our regular schedule, like giving online class, giving checking assignment and everything. So thank you so much, Ajahn. So thank you, everyone.
Thank you, sir, and thank you everyone for being present today.
Yeah.